My name is Will Simpson. I'm the Director of Communications at PolarisQB. We are the first company in the world to develop a drug design platform built for a quantum computer. We call this quantum-aided drug design. And today, two of our scientists, Dr. Ken Byler, our Director of Computational Chemistry, and Dr. Anna Petrov, will be talking with you about how we are using that system to research different types of protein-ligand interactions, protein-protein interactions. So we're very excited to present this to you. And without further ado, I will pass it off to Ken and Anna.
Thanks. I'll get us started. So I'm Anna Petrov. I'm a computational chemist here at Polaris Quantum Biotech. And thanks for coming to our webinar, Find Your Star series. So for the last 4 years, we've been working with partners and clients to accelerate their drug discovery timelines with quantum annealing computers. So we're based in Durham, North Carolina, and here's a photo of our whole team. Half of us are chemists and the other half of us are engineers, and we work together to translate chemistry into the language of quantum computing. So let's look at our agenda for the day. So how do we build libraries of preclinical drug leads? So we'll discuss how our approach complements other approaches to speed library design. How does the quantum computer aid in this process? And for that, we'll cover how we translate this problem into something that can be solved on a quantum annealer. And then we'll look at our successes. For that, we have some case studies, including single protein and protein-protein interactions. And then we'll have time for questions. So let's talk about quantum computing for drug discovery. So you're familiar with CAD, which uses conventional computers to find future medicines. We're using QUAD, which is quantum-aided drug design, and it uses quantum computers. The specific quantum computer that we've been working on is D-Wave's Advantage. While we're working with D-Wave right now, we also have a DARPA contract to expand our process to other architectures and to future machines. So why are we using quantum computers? Well, wet lab searches are limited to synthesized molecules that are on the shelf ready to be assayed. And those tests are typically limited to a few hundred to maybe a few thousand molecules. So you're solely searching a small chemical space, which can be appropriate, but really only towards the end of a drug discovery project. You can also add computational approaches like computational docking, but this is a brute force calculation, which is going to limit library size. So you shouldn't need a supercomputer to search even small libraries. Several of us at Polaris came from writing grants to get time on supercomputers. But at the end of them, like it still wasn't fast enough, and the results that we were getting were highly variable. So what about AI/ML? Because we're using a quantum computer and not AI, we don't need a big training set. So for that reason, we're not limited to pockets that already have a lot of information. The other issue here is that when you're looking for a new drug, it may be so different from existing drugs that are in your training set that it may be like any hits that you are actually looking for are going to be outside of the training set that you used. So because we don't need a training set, we also don't have the energy needs or have the carbon footprint that AI/ML has. So on top of that, it's also often a black box approach. So it may be hard to explain how you got what you got when you're using an AI/ML. So where do these approaches leave us? Wet lab, docking, AI/ML, and other traditional methods aren't moving fast enough to find medicines that we need today. So the timelines are way too long, often taking months to years and costing millions to billions. So we've just got to work together to pick up the pace to find new medicines. So we think of our approach as quantum computing for a modern accelerated drug discovery pipeline. So how do we build libraries of preclinical drug leads using today's commercially available quantum computers? Let's see how it works. So the goal of QUAD is to speed the identification of small molecules that may be future medicines. The input for QUAD is 3D coordinates of the protein and a small molecule in the binding pocket of that protein. So we see a protein here in blue and a small molecule in the binding pocket shown in purple. We pair this information with a massive chemical space of fragments, and these fragments are the building blocks for small molecules. So the goal is to find a good combination of fragments that can be assembled into small molecules that fit that binding pocket. However, chemical space is huge, so we use the quantum annealing computer to help search that space. So Quad uses the quantum computer to find combinations of fragments that can make small molecules that fit the binding pocket, are synthesizable, they're bioavailable, and they meet other parameters that are specific to your drug discovery project, such as being able to cross the blood-brain barrier. So the output is a custom library of candidate molecules in 3D conformations in the binding pocket. So with our library of candidates that were built for the pocket that you're studying, you can go ahead and synthesize these molecules and test them in the wet lab. So this approach beats the industry standard, which is based on iteratively checking small molecule libraries or designing the molecules by hand. So we have several white papers on this topic, including how we cut months off of library generation timelines. So at this point, I'll pass the mic to Kendall Byler, who is our Director of Computational Chemistry. He's going to talk about how the program works and some of our findings. And then after that, I'll present about a new feature that we've developed for a complicated target. So here's Kendall.
Thanks, Anna. Let me go into some detail about how the structures are generated. Quad starts by partitioning the binding site into subregions based on corresponding template features. It then searches a library of fragments with topological complementarity to the subregions and replacing essential features such as hydrogen bonds, polar interactions, and hydrophobic interactions. Synthesizability of final structures is based on a set of retrosynthetic rules. Finally, there's a geometry optimization at the end of both the ligand and the side chain residues for each candidate structure in the binding site. The way we do this on the quantum computer is by adapting the well-known knapsack problem to work with molecular fragments that are best fits to each subregion of the binding site that also obey a set of retrosynthetic rules that are applied as constraints. This would allow a medicinal chemist to readily synthesize the resulting candidate structures coming out of Quad. The formulation is a simplified scoring function that is solved as a quadratic unconstrained binary optimization. And we perform these calculations on a D-wave advantage system with over 5,000 qubits. Expressed in terms of graphs, the graph of optimal paths containing connectable fragments with good complementarity to the binding site are mapped on the QC, and the problem is solved as the system relaxes to the ground state. Since the system is described at the accuracy of a molecular mechanics force field throughout the process, we recover many structures at or near the Pareto front. That is to say, at the level the force field is still able to distinguish between better and worse candidates. Structures with good scores are then subjected to property evaluations to identify the best candidates from a pharmacological perspective. The diversity of structures generated by Quad thus results from trade-offs between the individual properties. So TPSA versus solubility versus shape complementarity, membrane permeability, etc. So how good are the Quad candidates? We characterize the performance of Quad through a series of studies that we published as a set of white papers. Here I'll talk about how we approached our evaluations in two such studies, one plain vanilla case study and another specialized mode of Quad we call best in class. So in this first study, we compared the binding affinities of Quad structures to those of known binders, as well as to those of decoys with closely related properties. Since we had to dock the decoys into the binding site, we used the same scoring function as our measure of affinity to arrive at an apples to apples comparison. As you can see on the left, Quad structures have better affinities than those of both the reference compounds and the decoys. We also compared the scores of structures designed by a computational chemist using current methodology. On the right, we see the same result. Quad structures exhibit better affinities versus those from conventional rational drug design strategies. Expressed in another way, this is a representation in terms of enrichment factors. We see on the left that Quad outperforms against physicochemically similar decoys, as well as those from rationally designed structures. When we focus only on those structures with affinities better than those of published structures, we see an enrichment that dramatically outperforms a conventional CAD approach. So are we just replacing fragments with very similar fragments and seeing what we want to see? Well, to test this, we compared the protein-ligand interaction fingerprint scores and max key fingerprint similarity values of Quad structures against those of the decoys. We can see that the quad structures more closely recapitulate the binding mode of the template. On the left, you can see versus the docked decoys. So the quad structures are binding in the same way that the template structure is, while having a relatively low structural similarity. As you can see in the middle, the average of that curve is about 0.66 Tanimoto score. So they're not very structurally similar to the template, but they are reproducing the binding interactions inside the pocket. In addition, the quad structures exhibit varying similarities to other quad structures in the output, as can be seen on the heat map on the right. The structures generated by quad are also subject to drug-like constraints through a configurable elastic Lipinski filter. Here we see the filters in action with the quad property values in red on the left and the reference structure values in right in blue. Template properties are on dotted lines for reference. This is how we can tune the output of quad to generate a synthesizable drug-like library of structures in an automated fashion. So how do quad structures look inside the binding pocket? The short answer is similar, but not too similar. We can see that a representative quad structure recapitulates the same binding mode on the right, but with significant bioisosteric replacements. The template structures on the left, the interactions exhibited by the template structures are retained. This is typical of the structures generated by quad, where 3D topology, hydrogen bonds, dipole-dipole, and hydrophobic interactions are replicated inside the pocket, referencing the template, although the quad structures are variations on this theme. At this point, I'd like to introduce quad's best-in-class mode. And to do this, I'm going to give you a brief story about the timeline and development of sorafenib, which was our test bench in this case study. In 1994, Bayer formed a team to develop new cancer medicine for late-stage cancers. And then in 1995, they started a high-throughput screening at Glaxo Wellcome, which took a year to test 200,000 molecules. In 1996, they started synthesizing 1,000 molecules, and after 3 more years, they found sorafenib and nominated it as a preclinical candidate. They started phase 1 a year later and started writing IP to protect that finding and received FDA approval in 2005. Altogether, it took 11 years to get sorafenib and patentable structures through the pipeline. Quad's best-in-class mode prioritizes the shape and pharmacophoric features of the provided template in its exploration of the chemical space that is closely related to the template. This mode can, in principle, be used to build or to break IP surrounding a lead compound. Here we use the crystal structure of sorafenib bound p38 MAP kinase as the starting point to explore the chemical space around sorafenib. So by functionally restricting the search space to the vicinity of the template structure in order to probe the boundaries of intellectual property, we find that there are clusters of locally similar structures, but that the set of compounds is actually rather diverse, as you can see in the heat map. Again, we used protein-ligand interaction fingerprints to assess the similarity of binding compared with that of the template structure. So focusing in on those that perfectly replicate the binding interactions as determined by the protein-ligand interaction fingerprints, we have 50 quad structures. And so these are fitting into the pocket in a what you would call a perfect fit. We can see that the structural similarity to the template for most of the compounds is actually pretty low, with local regions of higher similarity. With all of the structures replicating the binding mode of sorafenib, they have an average structural similarity of 0.78 in terms of their structure. So quad is not just replacing sorafenib with identical structures, although there are several close analogs. It is producing structures that might appear within or just beyond IP filing. So next, we did an IP search for these structures, and what we found was they exhibit an identical binding mode. 3 of these were described in either literature or in patents as MAP kinase inhibitors. Similar structures also extend out in chemical space from the template in a way that would allow one to describe related compounds in patent filing. Also to explore structures just outside of IP protection. This is useful, again, for constructing an IP moat and also potentially for breaking an IP moat. Next, Anna will describe a newly developed application of quad to PPIs.
Thank you. So we've been talking about typical quad projects, and now we're going to look at a new development, which is looking for inhibitors for protein-protein interactions. So here we have an example of a single protein in white and a small molecule ligand in the green pocket, and this is a typical quad project. We can work with many kinds of ligands in the pocket, including small molecules, peptides, unnatural side chains, macrocycles, peptidomimetics, and tool compounds that do not have drug-like properties. However, not all diseases can be treated as single protein problems. So for example, we've got the Ebola virus. So here we see the interaction between two proteins, the Ebola virus protein GP1 highlighted in green, and the human protein NPC1 in magenta. So this is unlike a single protein with the ligand that we have been using for quad. So instead of a small pocket, we see this large hydrophobic interface that is 3 times larger than the small pocket. So also there's no ligand in the pocket to use as a jumping off point for drug design. So this Ebola human protein-protein interaction is typical of other diseases caused by these interactions, such as many kinds of cancers, autoimmune diseases, and viruses. Also, PPI inhibition is considered by many to be the most difficult area of drug discovery. So we wanted to expand our system so that we can use the quantum annealing computer to design small molecules that inhibit protein-protein interactions. So here's a way to visualize what we're trying to do. Here's the interaction that causes the Ebola infection. We want to design a small molecule that can get in the way of these two proteins interacting. So if this is outside of your area, one way to think about it is you can't shake hands with someone if you're holding a baseball. So we need to design a small molecule, which is that baseball, to put into the hand, which is the human protein. And if the hand is holding a baseball, it can't shake hands with the Ebola virus. So this is what we mean when we say we're trying to inhibit the protein-protein interaction with a small molecule. So what do we know about this pocket, and what do we need to do to design a small molecule for it? So we know a lot about the pocket thanks to Wang et al. And many, many other researchers working on this problem. So here's the human NPC1 protein as a magenta surface, and the Ebola virus is shown as a green cartoon. So when we look at the parts of the human protein, we can see that there are 3 areas of interest. A hotspot, which is a super hydrophobic area that draws the proteins together. An area where 2 proteins interact, and also a base, which is a convex area below the interaction hotspot where a small molecule may be able to hold on. So it's like the baseball nestled in the palm of your hand. So one thing that's missing here is usually drug discovery projects have a small molecule template in the pocket to design off of. So that's the new feature of this project is we can put a tool compound into the pocket and then design off of that tool compound. So the first question is, where can we design into the pocket? Well, let's take a look. So when we just look at this pocket, it can be hard to look at this big interface and see where to design for it. So maybe we could try to design a small molecule for this area right here so that it blocks the interaction area and also it's alongside this hotspot. Or maybe it makes more sense to wrap it around the hotspot like that, and so we're hitting this interaction area and also the hotspot. Or maybe it would be easier to have it extend down into the base. Or maybe it can come over this way and reach into the base in this direction. So we wanted to make this program that builds a series of templates into the pocket, and this allows us to quickly generate options and test them. So we start with a fragment collection of 3,000 fragments. These fragments are of small molecules, and then we dock each of the fragments all around the binding pocket. And then we collect the fragments with the top scores, and we increase the diversity of the fragments by changing the functional groups and growing them into small molecules by combining the most favorable docked fragments. So we can also do hand modifications to these fragments to direct our options. And then we redock the new generation of molecules, and we do that over and over until the fragments grow into a diverse set of small molecules all across this large interface. So these small molecules we call Franken molecules because we're taking available fragments and then combining them into small molecules. We use this tool sequence. So we started with the empty protein pocket here, and then we created three Franken molecules to fit this pocket, and then we used these as the inputs for quad. So the nice thing about the Franken molecules is that we can use them as a jumping off point for design. We were able to generate a library of small molecule candidates that may be able to inhibit the interaction between the Ebola virus and the human protein. So let's take a look at the platform where we upload the protein and tool molecule into the pocket. So this slide is from our quad dashboard. We have a whole webinar on how to use it, from uploading the protein and ligand to downloading a library of small molecule candidates for your protein pocket. In our case, we'll upload the human NPC1 protein with the Franken molecule tool compound in the binding pocket. And then Quad will generate a fragment library for that pocket, where it will process the ligand and pocket and generate the fragments for the pocket. Then the job will be sent to the quantum annealer to find the combinations of fragments that best fit that pocket. And then it will calculate the properties of the whole molecules that it made, so that it can filter the library back down such that the candidates match the project design goals. And finally, we do a gentle minimization of the ligand in the pocket in order to resolve artifacts from the small molecule in the pocket. So what candidates did Quad generate for our Ebola protein-protein interaction inhibitor project? So we ran 3 Franken molecules through Quad. We first returned a library of 398 candidates in that pocket. And this GIF shows some of those 398 candidates. So when these small molecules are in the pocket, we don't think that the Ebola protein can interact with the human protein. So we also ran more libraries. Here's Franken molecule 2. We found an additional 61 candidates. And then for the third Franken molecule, we found 11 small molecule candidates. So although 11 may seem like a small number compared to the other libraries, these are pretty good candidates. They have better binding energy than the Franken molecule. And also we can run these back through Quad to make more. So how do you keep improving the Quad candidates that you get? We can run them back more to make the additional candidates. So we've got 2 modes for doing this, as Ken mentioned earlier. This is what it looks like when you use our dashboard to upload them. So you can upload your PDB structure, and then you can select your best-in-class mode or your first-in-class mode here. And with the best-in-class library, you can see the small molecule library has many clusters of similar molecules all along this diagonal. And so this mode allows you to use like a fine-tooth comb to search for similar molecules. Whereas in the first-in-class, the molecule library is much more diverse, and there are no clear clusters as there are over here. So here's a cluster, and then here you can see it's much more scattered. So this allows you to generate many options based off of the reference template. So another way to look at this data is to look at protein-ligand interaction fingerprints. So along the x-axis are the protein side chains, and each of the small molecule candidates is represented as a horizontal row. So each of these ones is a candidate. These interactions between the small molecules and the protein side chains are represented by this dark line here. So for the best-in-class, you can see clear columns of interactions showing how these small molecules have similar binding modes. Whereas when you look over here in the first-in-class mode, you can see that you don't get those same strong columns that you do in best-in-class, that it's interacting with the pocket in different ways, which may be what you're trying to achieve in your drug discovery project. So what are the next steps? We've also found that other protein-protein interaction inhibitors that we've made through this project have been active in wet lab tests. So for example, the industry standard is to synthesize 100 molecules and find between 2 and 5 that are active. And in a recent study, we synthesized just 30 molecules, and we found that 10 are active. So in addition to being many months faster than traditional methods, we're finding more molecules that look like viable preclinical drug candidates. So this is what Quad looks like from 1,000 feet. So the output is a library of 3D molecules in the binding pocket that are sorted by molecular properties, drug likeness, and pocket fit. The output from multiple different modes covers many design goals, such as using best in class, first in class. We also have a scaffold hopping mode, and we can inhibit protein-protein interactions. What do you want to do with your quad results? You can order and test them. You can test them from either a de novo library, or you can use quad to build within a commercial library, such as Liverpool Paraffin. And you can also do additional investigation of these results. You can explore quad output with QMMM, CAD, or other traditional methods. So we'll move on to our question and answer section. I've got a few different QR codes to share. So if you'd like to read more about quad libraries and strong docking scores, this QR code will bring you there. We've also got a good wet lab paper, a good white paper on recovering key groups with positive controls here. We've got a webinar with Maurice Benson, our principal quantum engineer, on the DARPA project that I mentioned. We've got a webinar demonstration of the quad dashboard. And then if you want to try the quantum annealer, we have a white paper on how to optimize a chicken and waffles order with the quantum annealer. So you can access that here. And if you want to talk about your project, or if you have a lengthy question that you'd rather discuss in a meeting, you can email us a question and we'll respond after this talk. And so with that, I'll open up the floor to questions.
Thank you, Anna. Anyone can raise a hand to ask a question and we will recognize you, or if you want to submit them by email. We did get a couple questions by email. I can start with one of those here to get us started. Is just a question that someone had of what other kinds of targets have we worked on using the quad system?
So we've used quad to explore some transmembrane proteins, GPCRs, and ion channels. This is ongoing work with customers. Basically, anything is useful. We can even use a clustered trajectory to use as a starting point for quad. So if you expect that your binding site has a large degree of flexibility in the pocket, we can do multiple frames from the trajectory as the starting point.
Another one that was submitted is whether quad is able to give us a 3D structure in the pocket, or whether it depends on additional docking for that.
No, these structures are generated inside the pocket, so there's no docking involved. It's a fragment goes into a subregion, is connected to another fragment, is connected to another fragment, and then the whole system, or the local system, is geometry optimized. So you get your quad library that you get back is 3D structures that fit inside the binding pocket.
One thing that I think people that we did see and we've heard often, and I don't know if Ken wants to respond to this or there are other members of our team here, is, and this could be a final kind of closing question, is what are the real advantages of using a quantum computer for this versus a CPU or a GPU or a lattice network of GPUs like other platforms may use?
That sounds like a Maurice question.
Is Maurice available?
Hey, no problem. Thanks, Kim. I'm Maurice Benson, principal engineer with PolarisQB. And the main reason we want to try to use quantum computers is they don't necessarily solve all problems well right now, but they do really well in trying to generate or trying to solve combinatorial optimization problems. So that's why we've leaned our technique into that combinatorial optimization problem. So that's the reason. That's like the quote unquote utility you get from using a quantum computer. It's the ability for it to solve those combinatorial optimization problems quickly.
We did have a couple questions in the chat. I see one here that says, is the protein kept rigid during the search?
It's in the process of answering that, but I'll do it here. Yes, the protein is held fixed during the search process and generation of whole molecules. And then each one of those that are candidates that pass the filters is then geometry optimized along with the side chains. So through the whole process, no, it's not kept rigid. But for the majority of the process, it is kept rigid.
And
And then
Ken,
another
maybe
one
you
from-
can talk, maybe
Oh,
you
sorry,
can talk
Shar.
a bit about using an MD and clustering.
Yeah. So if like I said before, if you feel that if you have evidence that your binding site is particularly flexible, I mean, we have looked at this also, is if we do a simulation of something in the pocket and we can take different frames from the trajectory and then submit those sort of separately as quad jobs and then collect the output and look for the intersection of those outputs to find structures or small molecules that will fit optimally inside the binding pocket, although it's as flexible as it is. I see one here that says, can quad B apply to ligand-based design? So completely ignore the binding site. Yeah, we have a way of turning off the contribution of the binding site. So just using the template, that's possible. We usually just run it with the protein as well because we want to geometry optimize inside a binding site. But in principle, it can be used for strictly ligand-based design.
Okay. Well, I think we're kind of nearing the end of the question and answer session. We would encourage you that if you're interested in finding out more about PolarisQB, don't hesitate to email a question using the link that we've provided here or reaching out through, you can find us on LinkedIn, you can fill out the form on our website. And we'd love to talk further with you if you have projects that you're interested in talking about or just to find out more about how we are using the quantum computing system to really revolutionize the way that drug discovery is accomplished. I think with that, we will close everything up for the day. We really appreciate everyone attending. We will have a video file of this available within the next couple of days. And again, if you have any other questions, please don't hesitate to reach out to us. Thank you to everyone for being here today.
Yes, I see there's another question here.
Are we open to partnership opportunities, right? So with folks who have connections within the pharmaceutical industry. I could almost answer this one, but I'll pass it to Shahar to close us up for the day.
Yes, we are very happy to think about partnerships. And please, Ernst, just either send us an email or we can contact you and we will be happy to have those discussions.